Welcome, everybody. So I'd like to introduce the webinar today. It's called Updates from ASCO. ASCO just took place a couple weeks ago, and we're happy to share updates with you. So during this seminar, all participants will be in a listen-only mode, and there will be a question and answer session at the end of the presentation using questions submitted through the Q&A feature. Please remember the information provided in this web seminar is not intended as a substitute for your physician's guidance and care. We would like to thank Genentech, Cogent, Decipher, Blueprint, IDRx, and Novartis for their support for our webinar series. My name is Sarah Rothschild. I'm the Executive Director of the Life Raft Group, and I would like to introduce our presenter today. Dr. Herbert Loong holds conjoint appointments of Clinical Associate Professor in the Department of Clinical Oncology and Deputy Medical Director of the Phase 1 Clinical Trial Center of the Chinese University of Hong Kong. He is also the current and founding convener of the Prince of Wales Hospital Adult Sarcoma Multidisciplinary Team. Dr. Loong obtained his medical degree with a distinction in surgery from the University of Hong Kong in 2003. He is a recipient of the European Cancer Congress Fellowship Grant, the American Society of Clinical Oncology Annual Meeting Merit Award, and a Hong Kong College of Physicians Young Investigators Award. You may have seen him partner with us from previous webinars, and we're really excited to have him here today to share updates with our truly global audience who's registered for this webinar program. So I'm going to hand it over to him to take it over. Thank you.
Thanks, Sarah, and good morning, good evening, everybody. So thank you for attending today's webinar. Let me just quickly share my screen. Here we are. Again, thanks Life Raft Group for this kind invitation and for the opportunity for me to be able to share some of the updates from ASCO this year with abstracts as well as discussions regarding the GIST. I'm Dr. Herbert Loong from the Chinese University of Hong Kong, medical oncologist by training with special interest in sarcoma, as well as lung cancer and early phase clinical trials. And it's always a pleasure to be able to share with the patients community of some of the updates that we've learned from these major conferences. And these are my disclosures. Quick disclaimer is the fact that we'll be sharing some of the slides that were presented at ASCO, and these are of their copyrights belonging to the original authors of ASCO. Certainly, this is being used by courtesy for this academic as well as patients' educational event. The choice to discuss the abstracts were made by my own assessment of their academic merit and the impact to the GIST community. This does not necessarily reflect the views of ASCO, of which I'm actually currently serving as a scientific committee member of in the field of sarcomas. So there are two main GIST take-home messages from ASCO this year. One is, in actual fact, there have been some updates from established as well as previously presented large randomized clinical trials in GIST. And we're also in the era of molecular profiling, and there was one particular abstract that was being picked out for poster discussion where it was a landscape analysis of the different GIST molecular signatures. Now, by way of background, and I'm sure the community already quite is fully aware of this, is the fact that certainly when we think about GISTs, which are progressing, we have various lines of tyrosine kinase inhibitors. The most established line, of course, is our first-line treatment, which is the use of imatinib, where we do get a fairly good benefit for our patients with median progression-free survival reaching about 19 months and a response rate of about 50% or above. Now, unfortunately, as patients are on this treatment over a period of time, there will be some who will have a disease progression and resistance. And in such situations, we actually have now established second-line treatment, third-line treatment, as well as fourth-line treatment with differing response rates, as well as differing progression-free survivals. Having said that, both in terms of what we see in clinical trials, both as well as the reports numerically, as well as our clinical practice, we do recognize the fact that these second, third, or fourth-line treatments don't benefit as many patients as we would see in the first line. And many of this have to do with how we're unable to fully separate out the different groups of patients who have progression after the use of imatinib. The larger phase 3 clinical trials many a times do see a dilution of these results because you're combining different patients or different molecular signatures into the same group. And I think at ASCO this year, what they've tried to do is look at the larger clinical trials and try to tease out or dissect out the different groups of patients in the second, third, or fourth line setting, and then to see whether or not we are able to identify specific groups of patients who respond better to one treatment or another. Now, the first abstract that I'd like to discuss is this particular one. It's actually in the form of a poster that was presented by Dr. Robin Jones from the UK in collaboration with all the investigators in the Intrigue study, with the senior author being Dr. Michael Heinrich. And this is actually the overall survival as well as the long-term safety data of patients who were treated with ripretinib, as well as with sunitinib in the Intrigue trial. And it's just to remind the community of which trial this actually is. The Intrigue trial is a phase 3 clinical trial which involved patients who are of adult age across the world in both Asian, European, as well as North American and South American sites, looking at patients who are on second-line treatment, who initially had imatinib with known mutational status of KIT exon 11, KIT exon 9, KIT PDGFRA wild type, as well as other KIT PDGFRAs. Now, patients are stratified based on all these findings. So what that means is patients who have these were equally distributed between the two groups. And these patients also had to be intolerant to the use of imatinib. This was a 1-to-1 randomization study where half the patients are allocated to ripretinib. The other half was allocated to sunitinib. And there was also no crossover option at that time. And the primary endpoint is progression-free survival. Now, you would recall in actual fact that in terms of the data of this particular trial, looking at survival for the entire group, it was found to be not significantly different between sunitinib or ripretinib, and ripretinib was not able to outperform sunitinib. So in terms of a clinical trial, it was considered a negative clinical trial. Having said that, we were able to find previously that ripretinib is just as good as sunitinib. And in terms of safety profile, it's better tolerated. And in actual fact, the use of ripretinib is in the second line setting is already currently in the NCCN guidelines, which is the guidelines that most physicians, at least in the US, would follow in terms of being able to prescribe these treatments for our patients. Now, going back to this clinical trial and some background about it, it's a very large international trial. Over 450 patients were recruited. 444 of them received treatment. Overall, 51 of the 444, which is 11.5%, remained on treatment at the time of reporting. And this is based on results in September 2022. There were patients, of course, who had to discontinue from treatment. And most of the time, it's to do with disease progression. And what they found was that there were fewer patients who discontinued treatment because of adverse events. And in the ripretinib group versus the sunitinib group, and the similar results were observed in patients who had KIT exon 11 populations. On the right-hand side is actually a table showing the anti-cancer therapies of which patients received after they progressed on either ripretinib or sunitinib based on the clinical trial. What you can see is that, and let me try to get the pointer out so you can see it better. What you can see is the fact that in most of the patients who was initially recruited into the ripretinib group, upon disease progression, about 69% of these patients received further treatment. The majority of it were receiving sunitinib. There were a couple of patients who received other TKIs. The majority of the patients in the sunitinib group actually did receive other TKIs as well, including ripretinib. But interestingly enough, this was actually not the majority. And most of the patients actually had the standard treatment of third-line regorafenib, possibly before going into other types of treatments. Now, I think this particular history of what treatment followed the investigational treatment is important, sometimes when we look at the overall survival of our patients. Now, these are the results of patients who were in the trial. On the left-hand side are for patients looking at their overall survival. And in actual fact, on the right is looking at progression-free survival. And without going into too much details, what you can really see is the red line are the patients who were on sunitinib, which is the control group of the trial. The blue line are the patients who are on ripretinib. And you can see that the two curves pretty much overlap and are quite intertwined, really showing the fact that there is no clear statistical difference in terms of the survival of patients, both in terms of progression-free survival or overall survival. Now, I think the crux of the discussion between the use of ripretinib or sunitinib in the second-line setting really rests on the side effect profile of these two drugs. And this is an updated data set, meaning that there have been patients followed for a longer period of time. And I think the conclusion of this particular abstract reconfirms what we have known initially, which is in actual fact, patients who are on ripretinib found the drug better tolerated and were likely to continue with the treatment, unlikely to discontinue, unlikely to dose reduce, or unlikely to interrupt the treatment compared to patients who are on sunitinib. And what you can see here is actually exemplified by the table here on the left. And in actual fact, specifically in terms of the side effects, what we find is that ripretinib is associated with less hand-foot syndrome, less blood pressure changes, less diarrhea, and less mouth ulcers. Now, again, many a times we would use the different grading of toxicity. And as clinicians, sometimes we think that a side effect has to be severe and is only severe if it is at grade 3, which is when it actually leads to potential hospitalization. Having said that, I think we're more and more mindful of the fact that even a low grade or say a low severity hand-foot syndrome, which is associated with the treatment for a long period of time, is something that patients do not find tolerable. So I think it's very important for us to recognize this as clinicians as well as investigators in the community. And many a times we actually get feedback from our patients, such as yourselves, and I think this is also very important. So I think if we're able to find and establish a drug which has similar efficacy but less side effects, that's also already a win for our patients. Now, the second abstract I would like to discuss also originates from the Intrigue study. So the very same study, but looking at different data. And this is a poster that was presented by Dr. Jonathan Trent with Dr. Sebastian Bauer as the senior author, looking at the use of circulating tumor DNA in patients who were recruited into this clinical trial, and specifically for patients who actually did not have detectable circulating tumor DNA. Now, we actually talked about circulating tumor DNA last year at this very same meeting, and I think it probably necessitates a bit of a background refresh in terms of what we're really talking about. And this gives me a good opportunity and segue to go back to some of the older slides that I presented actually at the same meeting a year ago. And what is circulating tumor DNA? Well, circulating tumor DNA are actually cell-free DNAs, which are small DNA fragments in the blood circulation. This actually happens when these tumor cells are actually dying, and then they will release their genetic material or their DNA into the bloodstream. In actual fact, circulating or fragmental DNA is found in healthy individuals as well, but that typically is from their own cells, specifically their own blood cells or white blood cells, etc. Whereas in actual fact, in cancer patients, we also find specific circulating tumor DNA, which actually originates from the tumor. Now, circulating tumor DNA is quite different from normal DNA because they're actually smaller fragments. So it is possible for us to identify the different types of DNA that's in the circulation. And that is the crux of us being able to use this technology to assess tumor growth. And in actual fact, the half-life of these circulating tumor DNAs are actually relatively short. Now, circulating tumor DNA is a hot topic and frequently used now for testing in terms of being able to obtain the genetic material from the patient's tumor. Historically, we would use tumor tissue assay, which essentially is a biopsy, which is invasive, which actually potentially have risks. We're only able to obtain a small tumor region in the big tumor. And there are situations where, in actual fact, the location of the tumor would mean that you cannot actually get the biopsy. Now, when we get these biopsies, especially when patients are on treatment, the really main aim is just to get the circulating tumor DNA, or just to get the DNA material. Then we could then use it to do various molecular tests. And what we found is that circulating tumor DNA is certainly less invasive. And the great thing is you are able to do it over a period of time serially. So you can then monitor the patient's progress whilst they're on treatment. There are some specific requirements where this blood that's been drawn from the patient has to be placed in special tubes, and then they do have to be processed within a certain period of time. But these logistical challenges are actually improving as we speak. And again, we do use circulating tumor DNA in quite a lot of other tumors, including lung cancer. So I think the technology really is already there. Now, the applications of using the circulating tumor DNA can be very far-fetching. Traditionally, we would use it for molecular profiling. We want to know which particular type of GIST, for example, exists in the patient at that particular time. Is it exon 11? Is it exon 9? And by using the circulating tumor DNA, we can then establish this. The slides here are examples of lung cancer, but the same can also apply for GISTs. One of the other great aspects of the fact that circulating DNA can be assessed serially over time is we could use that to monitor treatment response. There is now improving techniques as using circulating tumor DNA for early cancer detection, and in actual fact, also the detection of what we call minimal residual disease, thereby hopefully being able to predict which patients will have disease recurrence or disease relapse early and be able to address and treat them earlier. Now, going back to the abstract that was presented, again, this is based on the Intrigue study, which is the phase 3 second-line randomized clinical trial between ripretinib and sunitinib. This is the exact same trial that we talked about previously, but we're now talking about the component or the translational research component, which looked at circulating tumor DNA. And essentially, patients at the time of randomization had a vial of blood drawn, where the blood is sent for a Guardant 360 circulating tumor DNA analysis. Now, within the group of patients who are within the clinical trial, again, 453 patients were randomized. Out of that, 362 patients had their circulating tumor DNA analyzed, and about 82 of them actually had the circulating tumor DNA being non-detectable initially, and 280 of them were detectable. And these were the groups of the patients in the different treatment arms with these characteristics. What we found was that the patients who actually had circulating tumor DNA non-detectable, ND, were typically younger and also had smaller tumors than patients who actually had circulating tumor DNA, which were detectable. And that does make sense in the sense that if you have a larger tumor, there's a larger chance that there will be tumor shedding in terms of the DNA material entering the bloodstream. Larger tumors are also more prone to necrosis, and that also accelerates the tumor DNA being entered into the circulation. Now, these are the results looking at the survival of patients who are in the clinical trial based on whether or not they have circulating tumor DNA detectable or non-detectable. The red line are the patients who actually did not have circulating tumor DNA that was detected initially. So the thought would be these patients probably may have smaller tumors or less tumor shedding tumors. And on the right is the overall survival. And what you can see is the fact that patients who actually did not have any circulating tumor DNA detected when they were recruited into the clinical trial seem to fare better, statistically significantly as well, over patients who had circulating tumor DNA being detectable. And we also see a difference in the response rates of patients, whereby in actual fact, there was a higher chance of the tumor shrinking if you actually had circulating tumor DNA that was not detectable upfront versus there being detectable. Now, this brings the trial to a further step of analysis, looking at whether or not circulating tumor DNA can help differentiate groups of patients who may or may not benefit from treatment. And with the two data sets in mind, including what we looked at in terms of the circulating tumor DNA, as well as an update in the survival data, it has led to the other clinical trial, which is currently running, which is the INSIGHT trial, specifically looking at patients, again, in the second-line setting, but the ones who have originally KIT exon 11, as well as the presence of a resistant mutation 17 or 18. Now, this study is currently ongoing, and it's a 2-to-1 randomization between ripretinib and sunitinib. We actually had this being presented as a trial in progress poster at ASCO this year. We're still looking forward to the readout of these results in upcoming meetings. But I think with some of this knowledge that we've gained from the prior two abstracts, as well as what we've learned last year, we're hopefully being able to fine-tune the way we approach these diseases and not looking at just as a big group of a singular disease, but there are different diseases, especially when they have been given initial treatments and they've developed this resistance, and to be able to hone in on specific groups of patients where there are very similar molecular characteristics and using one particular treatment for that particular group of patients, hopefully we'll be able to address the outcome and reduce the issues of heterogeneity. Now, the last abstract I want to just highlight to everybody, which was presented at the ASCO meeting this year, is a poster that was selected for poster discussion. Again, this was presented by Dr. Jonathan Trent in collaboration with quite a few investigators who have been using Caris as the molecular profiling provider for their patients. The senior author was Cesar Serrano. Essentially, this particular piece of work really addresses the issue of the molecular heterogeneity of patients and their GIST tumors. So going into detail, in actual fact, this is an analysis of 941 GIST patient samples that Caris actually has received over the years. And within these tumor samples, there were patients who actually had some DNA profiling, also some with RNA whole transcriptome profiling as well. And what you can see is on the right-hand side, the demographics of the samples that were recruited, including what you would expect. The median years in terms of age was 64, a fair balance between male and female. And we included patients who actually had biopsies of the primary tumor, as well as done those which actually were of the metastatic sites. They specifically wanted to look at the gene expression signatures of these particular tumor samples, as well as the tumor microenvironment. And essentially these two potential flags or bookmarks really do help us understand whether or not these patients may or may not respond to treatment, and as well as possibly an influence in terms of the survivals as well. Now, this is a relatively busy slide, but just to highlight the fact that we had within the study, this is what you would expect in terms of a practice of GIST patients, where the majority of the patients actually had KIT mutations. There was a small number, and this is almost exactly 10%, or sorry, 8.5%, which actually had PDGFR alpha mutations, as well as 10.5%, which were non-KIT, non-PDGFR alpha. So this is quite representative of what you would see in a single large GIST practice, and this percentage of distribution between the different molecular types is also what we found in this larger cohort. There were presence of primary mutations as well as secondary resistant mutations, the usual players being, say, exon 13, 14, 17, and 18. And I think the main purpose of this particular slide really isn't to go into the nitty-gritty details, but just to highlight the fact that we do see presence of other alterations or other mutations aside from the driver mutation, which is KIT. There are some which are more common than others. And this you can tell by the thickness of some of these lines. And if these bars are thicker, it means that they are also present in those samples. And the thicker they are, the more of these samples actually also carried, say, RB1 as well as TP53 mutations. Now, this also looks at the microenvironment, which is the not addressing the tumor cells themselves, but the neighboring structures, the neighboring tissue that's associated with the tumor cells. And specifically, we believe that the microenvironment has strong interplay with the tumor itself. And in actual fact, that can influence whether or not the tumor behaves indolently or behaves aggressively, or whether or not they may or may not be resistant to treatment. And essentially what you can see is there does seem to be a separation where there's more red here and more blue here. And the more the colors separate, it really shows the fact that within these samples, there are distinct groups where it is believed to be less abundant in terms of some of these inflammatory aspects, and some which are more. And I think this helps, again, us understand the fact that not all GISTs are the same, and that particular GIST tumor from that particular patient may be in one group versus the other. So targeting, for example, tumor microenvironment in terms of treatment approach may work for some patients and not for some other patients. Now, I think the very last abstract was a bit technical, but we could certainly go into finer details and look at it more in a more detailed manner. But I think the bring home message I wanted to take across, at least from the last one, as well as the first two abstracts, is the fact that we are having advancements in our field. And certainly we are seeing maturing data that was presented from earlier conferences. We have established again that GIST is a heterogeneous disease with varying clinical behaviors, and the further advancement of GIST management needs to be multinational, multidimensional, and multidisciplinary. All these three abstracts certainly are of very large GIST populations. The clinical trials are global clinical trials. Even the CARIS data set are from physicians as well as patients from around the world. And I think in situations with these rare diseases, we really have to work together to move the needle together as well. And a bit of a segue before I end is the fact that we presented the GIST data just now from ASCO. Admittedly, it's been relatively thin this year because we actually had quite a lot of GIST information that was presented at last ASCO as well as last CTOS. But don't worry, in actual fact, we are only just around the corner from our next large sarcoma gathering, and that is the CTOS meeting. This year, it's going to be held at the end of October, early November, and this would be held in Dublin, Ireland, where traditionally we see about 1,000 to 1,200 attendees. This is where we, as a sarcoma community, really get together and share some of the knowledge as well as advancements that we've made in our labs or in our clinical trials within the year. And really, it's a big symposium, yet small enough for us to really interact with each other and to bounce ideas off. And specifically, I wanted to highlight that we do have a GIST session that's dedicated specifically to GIST abstracts, and this will be presented on Saturday, November 4th. Now, CTOS is a meeting which is held physically, so do come if you can. But if not, all the presentations are recorded, and there will be a chance for you to access this information later on, as well as the posters also being uploaded into the website. So do follow us as well in terms of social media. Many a times we have very keen colleagues who are posting as well as tweeting throughout the conferences. And the great thing about these tweets are, they come straight to your iPhone or your device, and many a times also includes their expert commentary as well. And that really helps understanding the disease field. And that's where I'd like to end. And this is our group in Hong Kong and doing mainly GIST education as well as just patient management, not specifically to GIST, but also to sarcomas in general. And certainly if you ever have the time to come visit us, do let me know and I'll be happy to introduce you to our entire group as well as to show you around. So thank you very much. And thank you, Sarah, for this wonderful opportunity.
Thank you so much. For those who've listened, Dr. Loong has given some insider information. If you want to follow the latest updates, if you join Med Twitter, you will get that in real time. It's a nice way to get feedback about what's happening in terms of advances in the field from the doctors. But the presentation was excellent. And I think that you were able to explain it very clearly, some of these more difficult to understand concepts and some of the medical jargon. I think it was very well explained. So thank you for that. We do have questions that have come in, so I'm going to go straight to that. Okay, so the first question is, what are your thoughts on the recent Intrigue ctDNA data in second line? How often is ctDNA testing used in GIST? And do you see this changing in the future?
Yeah, that's a wonderful question. And I think ctDNA certainly is a technology that I think we will continue to employ and also increasingly employ in GIST management. Now, specifically to the data that I presented, which was the abstract that I presented, and that's the question as well. The ctDNA analysis for the Intrigue trial was done retrospectively. So what that really means is the patient's treatment were not influenced by whether or not they had ctDNA detected. What we can see specifically in the abstract that was presented is patients who actually have detectable ctDNA were possibly patients who had more aggressive disease or more resistant to the treatments, and therefore actually they did not fare as well. Now, I think the promise for ctDNA is not really to identify which group the patients would be in terms of their prognosis, but more so in terms of helping us guide treatment as we have more and more new targeted therapies becoming available, which have different characteristics. So what we have found certainly is last year with the avapritinib data, and this year, during the year with the Intrigue data, looking at ripretinib, as well as now an update as well, is the fact that the different molecular signatures of the patients do lead to different outcomes. And some of the TKIs that we use are better in some molecular signatures than others. Now, honestly, right now, there's still not that many treatments to choose from. So whether or not knowing the ctDNA and knowing the molecular signature may or may not make a difference. But as we further advance and as we have more treatments, as we now are able to say design tailored drugs which possibly address exon 13 and 14 specifically, and not 17 and 18, which is done already quite well by ripretinib, that's when ctDNA comes in, because then we're able to monitor these patients over time and to choose the right treatments for them at that particular juncture. Now, this is all conceptual and conjectural. We don't actually have solid data to say that using the ctDNA and guiding treatment will lead to better outcomes, but I can see the way the technology is going to advance. We're doing something very similar for lung cancer already. I think that's where the field is going to.
I'm going to add a few questions that people ask regarding circulating tumor DNA. So is there any indication or advantage from what we know now to do routine circulating DNA in patients who are on imatinib, who are stable and show no recurrence or resistance after resection surgery?
Yeah, I think right now, based on the data specifically for GIST, we don't have a clear indication as to why we would like to do that specifically for patients who are very stable and tolerating the treatment well with no indication of recurrence or progression. Again, we would potentially use that as one of the ways of monitoring patients. I mentioned the issue of what we call minimal residual disease. We are actually employing this technology now specifically, has always been doing for some of the blood cancers, but now also for lung cancer. a GIST does not seem to be a disease which has a lot of tumor shedding, which you can pick up in the blood. But I think with some fine tuning of the technology, one day we will see that. But right now, it's not a routine practice.
Do you think that people are now kind of getting confused between the plasma level testing and the ctDNA? Like, is there maybe
It's possible. Yeah, I mean, at the end of the day, they're both blood tests. Plasma level testing is drawing the blood, looking at the level of imatinib that's in your blood to see whether or not it satisfies a particular blood level. So the worry is, as time progresses, there's not enough imatinib in your body, or you're not metabolizing the imatinib the same way as what you would expect, and therefore the dosage of imatinib may not be adequate, and that may be one of the factors which may lead to not so good of a response, and you may want to adjust the dose of imatinib. Having said that, I believe those studies have been quite controversial in terms of their findings, and I believe this approach has fallen out of favor over recent years. Also, it's to do with the fact that we have other drugs now available. So if you're progressing on imatinib, of course, we have second-line treatment, third line, as well as fourth line. And even if drug level is one of the factors that may be associated with good or poor response, there are many other factors that come into play. And I think the development of resistant clones, such as different GIST mutations, different molecular signatures, would trump whether or not there are level differences. Okay. We don't do the levels here in Hong Kong. I'll find it quite hard to find a lab who would be willing to do it for us. So so for us, there's less of a confusion. But I do understand that in North America, it was a fashion, it was a fad some years ago.
Right.
And people were checking
right.
that.
And then the Dutch do it on a routine basis. Yep. Isn't there in Switzerland, some certain European countries, I think they do it on a routine basis. But
in actual fact, we were some years ago, we were talking with the Dutch colleagues about whether or not we could look at imatinib drug levels through saliva instead of a blood draw. So, but the project never materialized.
Right. Somebody also wanted to know how far away we are from using ctDNA for adjuvant use.
Yeah, so I think it sort of echoes one of the previous questions, whether or not we can use ctDNA to detect minimal residual disease, or patients who have detectable ctDNA, say even before surgery, do they necessarily also have a high risk of recurrence? And in those, should we be treating them adjuvantly for a longer period of time, or versus if they were still considered low risk, should we still treat them based on the fact that they have ctDNA detectable? I think there are many parts of the puzzle which we have not quite solved yet. A lot of the pieces are moving. We're generating new data. And I think the crux also has to do with how good the technology of ctDNA is. And these advancements are made dramatically over a shorter period of time. So this year, at least the conclusion is ctDNA is what we call prognostic. So if you have it detectable, what that means is the chances are the disease is more aggressive, you're not going to respond as well to treatment. In such situations, we should be more wary, and we should be making sure that these patients do come back for their regular follow-ups, on time, they get their scans on time. But we're not in a position to take a different course of action for these patients because we don't have that prospective data. But who knows? I think in 1 or 2 years, we may see new presentations showing the fact that ctDNA can actually help influence treatment.
I mean, just in this past year alone, I mean, it's just ctDNA has like been the big focus over the course of all these conferences. So I think it's just even going to like escalate from here. So more to come.
And again, it's not only for GIST as well, for many other tumors, that's a common theme. So I think we are going to see some further advancements in the next couple of years.
Okay, so does inclusion of ripretinib in the NCCN guidelines for second-line GIST patients intolerant to sunitinib change your practice? Does the safety profile of ripretinib support sequencing ahead of sunitinib? Good question.
That's a great question. I think it really depends on the physician themselves. I would think, in all honesty, having that opportunity to use ripretinib earlier on, and to be able to avoid the toxicities of sunitinib is definitely something that's very attractive to our patients. It also depends on where you practice, whether or not the drug is in the guidelines, whether or not it's reimbursed. but at least in terms of the data that we're seeing, it does appear that ripretinib and sunitinib in terms of overall efficacy is similar. But certainly, again, reconfirming that ripretinib has a better safety profile. Now, the INSIGHT trial that's coming up specifically now looks at a particular alteration group, exon 17 and 18, which we do believe ripretinib will perform better than sunitinib. At least that's the hypothesis. But the confirmatory data is not out yet. So we have, again, retrospective data based on a large Intrigue trial that patients who actually had secondary mutations of 17, 18 do better with ripretinib than sunitinib, but we don't have that solid data prospectively. That's one of the reasons why I believe ripretinib is in the second line guidelines for NCCN, so based in the US, but the European medical agencies have not accepted that as an indication for second line. For me, honestly, if the patient can afford and is able to have access, I would consider using ripretinib. You know, we are all quite aware of some of the side effects of sunitinib. It's not an easy drug to take. Even if the patient can tolerate it well, the dose interruptions and other subtle side effects is not something that I would like to see in my patients.
Thank you. Thanks for always keeping that in mind in terms of the side effect profile and the quality of life of patients. Have there been any barriers to access Sutent in second line in terms of affordability for patients?
I think the barriers are starting to come down.
It went off patent in the United States. Just
that's right. Yeah, so I think that's one of the ways the barriers have come down. It's gone off patent, so there are biosimilars or generics possibly available. I think in Hong Kong right now, Pfizer still holds the patent here, so we're still using brand sunitinib, but they've also made adjustments to their prices. It's been entered into, at least for us, a reimbursement scheme, so they had to lower the price. But I think the access of it, it depends on where you are living, in the sense that whether or not generics can also be registered and used. And that, I think the onus actually is on the drug company, whether or not they're willing to register that drug in your jurisdiction, because they have to go through the hoops and hurdles of FDA approval, etc. So I'm not too familiar with the situation in the US. That may be the case.
Sometimes I feel like we've seen it's taken a while for the generics price to come down. That happened with imatinib. I'm not sure if that's happening yet with sunitinib generic. It might just take time as more manufacturers enter the market.
Right. But there's certainly generics available. So I've seen patients with generics. Yeah.
Okay, so you didn't discuss this today, but somebody wanted to know, what are your thoughts on data from the PEAK study with bezuclastinib plus sunitinib?
I think the data is promising, but the trial is ongoing, so that's why I haven't discussed it today. It was presented, some of the initial results in the phase 1 portion was presented as a poster by Dr. William Tapp at the conference. I think that potentially is one of the ways for us to tackle the issues of addressing resistance early and having a combined use of the two drugs. At least based on the earlier phase data, we do see patients responding longer. So probably a little too early to go into conclusions, but my suggestion is if you're in a situation where you're about to start second line treatments with sunitinib, and if there is a clinical trial option, which is the PEAK study that's actually actively recruiting patients from around the world, we're in Hong Kong, actually, we're starting our recruitment next week for this particular clinical trial as well. So do talk to your physicians to see whether or not there's a clinical trial option that's available for you.
Do you want to just like the PEAK study has a different type of structure than other traditional trials? They don't call it a phase 3, they call it a part 2. Do you want to like maybe explain to patients when the trial designs become more adaptive and why that's happening in the GIST space now?
Yeah, I think it's a common thread in terms of not only GIST, but in other clinical trials as well. So traditionally, when we do clinical trials, it'll be broken down into the phase 1 trial, where small number of patients, we really want to look at the side effect profile and the safety profile, and whether or not it works well or not well, whether or not it's safe. Then we move on to a phase 2, where specifically in the phase 2, we want to look and see whether or not there's any good effects on the patients. And a phase 3 trial is a much larger trial, many a times with several hundred patients, many a times internationally. And I think the most important part is it's a randomization between the standard treatment as well as the treatment in question to see whether or not this is any better than the original treatment. Now, originally, the way we develop drugs, it's very compartmentalized. So you do a phase 1, and then you do a phase 2, and then you do a phase 3. And only if your phase 1 is completed, then you would start and consider doing a phase 2. And only if that's completed, you do a phase 3. And then also the regulatory authorities, such as the FDA, will say, well, I need a phase 3 trial before I let you register a drug. So it used to be very pragmatic. And the problem with that is many a times, especially in situations where we're dealing with rare tumors, it's very difficult for us to have that number of patients to be able to fill up these trials, to be able to generate the data. And most of the time, from a phase 1 to a phase 2 to a phase 3, in actual fact, only about 10 to 15% of drugs actually get to a phase 3. Many a times they actually have to stop after a phase 1 or a phase 2, either because there really isn't good efficacy, or sometimes it's just also has nothing to do with the drug itself. Maybe the drug company is out of money, they're unable to further develop. And what happens is we see a lot of patients left hanging in the middle. We are not able to consolidate that data between the different phases. And essentially, we're also potentially causing harm to a lot of patients, because a lot of patients are put into these trials and we can't be sure whether or not it's efficacious. Now, in the PEAK study, it follows a newer design where the phases are a bit more blurred. There is not that compartmentalization between the phases. And a lot of the data that's generated in the phase 1 portion can be used in terms of a phase 2. And the different endpoints are also rolling. So what that essentially means is we are able to get to the endpoint of whether or not to see the combination in the PEAK study is better than sunitinib earlier in terms of time frame, because we don't have to go through the different phases. All we need is a change in the way we analyze the data in terms of the biostatistics, in terms of the study design. And I think most importantly is we don't need that a high number of patients to reach our goal, which means that there'll be less patients randomized to the control arm, which we suspect would be less efficacious than the investigational arm. Again, we will not know all this until we finish the study. So there's a chance that when we finish the study, we realize that it's not any better than sunitinib. But at the same time, I think we're hopeful that it is. And I think especially when the control arm in this particular trial already includes sunitinib, it's an add-on to sunitinib. So I think in this such a situation, it is reasonable for us to try to compress the investigation of it, as long as we finish the part 1, which is to look at safety, and we realize that the safety profile of these patients, when we select the right dose of the investigational portion, is acceptable.
Thank you. That was an excellent explanation. And also, I feel like it gives us hope in terms of looking at future clinical trial design differently in the GIST community, which is great. Any new therapy on the horizon after 4th line?
I mean, there are certainly other drugs that's being investigated. We've seen some of those in previous conferences. They just haven't presented it this year. I think one of the other aspects is the multidisciplinary aspect of GIST management, which I have not been able to highlight this year because there were not that many abstracts talking about this. But many a times, we always follow one track. We go to the first drug, second drug, third drug. But I think for our GIST patients, what we really need to do, and we really should do, and a lot of us are already doing, is each time when we need to make a treatment decision, it should be multidisciplinary. So there are situations where, say, the tumor is growing and we might need to change to another treatment, but maybe it's just one or two tumors which can be addressed through other means. So not drugs, but maybe a surgery, maybe some sort of local ablative therapy. And I think the advancements in terms of the treatment outcome for GIST patients has more to do with the fact that we are looking at tackling the tumor in a multi-pronged approach, as opposed to just a one-track mind of drug. There have been abstracts that's been presented looking at this, looking at the metrics of this, and I think it also confirms the fact that that really is the approach. So my conclusion to that, or my answer to that particular question, is the fourth line treatment or beyond fourth line is to bring your surgeon back in and for us to reevaluate whether or not there are other options besides drugs, because at the best, to be honest, drugs are able to keep things at bay, but we're not seeing regression of the tumors. And what we really need may be some sort of tumor removal at the right time.
Speaking, that very well said. So I don't see any other questions that have come in. I'm gonna leave some space and time if anybody else wants to add any questions in for our speaker. There's still some time left in the session if you'd like to ask. I think if anybody can take something away from today, it's great that there's still interest in terms of clinical trials and studying this rare disease further so that we can help improve patient outcomes for our disease community. So not seeing any other questions come in. So I guess then we can conclude today's session. I just want to say it's such a pleasure partnering with you with these webinars, because you're so tuned in to what's the latest science. And it's great that you're actively involved in these scientific committees that are part of these congresses. So having you be able to relay that message to the patients is really important to us. And we are going to be, Life Raft Group will be at the CTOS conference. We are happy to represent the GIST community there and join in on that session, and also meet the experts from all around the world to hear the latest advances. One person did, sorry, one last thing before we leave. I think somebody did mention that there's a drug that showed some efficacy in SDH that came from China. I think that was probably from sdh. It was not featured as a presentation, but did you have any background that you wanted to relay to people about that study?
Yeah, it was, if I remember correctly, it was a poster discussion. I can't quite remember which category. So I didn't actually highlight it during this talk, mainly because I think we covered it last year. It's actually just an update of the trial that they were doing in China. I think certainly sdh-deficient just is honestly a completely different kettle of fish in terms of the biological behavior. And I believe that there was efficacy in that particular clinical trial. We could certainly circle back later on where we could discuss the finer details. But yes, that was one of the abstracts that was actually highlighted, presented at ASCO.
And you know what? Just Awareness Day for everybody. Just Awareness Day is actually taking place on July 13th. So I hope everybody who's listening can do something to raise awareness, social media channels, friends and networks, to just talk about the disease. But we are having a panel discussion that will be discussing SDH, and that could also be brought up there too. So we can explore that the findings from that study further. So I'm actually going to just conclude the presentation today. Thank you for everybody who participated. And thank you, Dr. Loong, for your presentation. And with that, I just want to wish everybody a wonderful rest of your day.